Karen, did Noelle get added as a developer yet?
Yes, she did.
Okay, good. So she's been entering the question. Well, good afternoon, everyone. I apologize for being a bit late. I had to attend a meeting, actually the university scheduling committee meeting, trying to figure out how we're going to deal with when we offer classes and when anybody can park anywhere within reason. And I was a little late for that meeting, too, because I couldn't find a parking spot. So, really is a fantastic change that's occurring with UTSA, and they expect even 2,000 more students next year, not the next semester, but next year. And we just can't build parking garages and parking places fast enough. And if we just spread classes out, then there's no guarantee that somebody that comes at 8 o'clock is going to be gone by 9 to free up that parking spot anyway. They'll just be here, traffic jam an hour longer, an hour earlier and an hour longer. So it really is quite a challenge. The first announcement I'll make today is that we will have lecture on Monday, and that means then that the exam will be offered completely online as of the end of the lecture on Monday. So in order to keep a pace with things, this is the mechanism that we're going to transition over to. So after the class, if this room is available, you'll be able to go ahead and take it here, or we'll have it available for a period of time, and it will be available to you to take 2 times, and I hope that everyone will have the opportunity to get really good grades, and we won't have to worry whether we average or take the highest. We'll assess the performance before we decide between those two. But it definitely will be offered just online, and starting Monday and very likely through midnight on Wednesday. Correspond through the WebCT email if there's any conflict for you based upon that intended schedule. So let's catch up on number 16 here. So this is the slide that we left off with. We identified cartilage, the different cartilages within the first and the second arches. These have unique names. They are called eponyms. They're named after individuals that identified them and studied them and reported it initially. And it is the Meckles in the first and Reichert in the second. And we'll follow the development of those into key structures in this area, as well as some reflection on the facial development as we have been following. So quickly to review, 1, 2, 3, 4 arches. The fourth arch isn't extremely well developed. The fifth and the sixth are really vestigial remnants. And each arch contains the vascular bundle, the artery being the only thing shown, neurovascular, the blue coloration is the nerve, the darkest red coloration is the muscle. So the neural crest cells that moved into this area differentiated into mesenchyme that produced the musculature and all of the connective tissue basically that will appear in this area, including the cartilage that will become part of the skeletal system. But the collagenous fibers in the extracellular matrix around the blood vessels, any collagen associated with nerves in the area, although not the actual myelin nerve cells and the sensory, those come from the straight out neural crest cells. And any of the tendons and ligaments that might be in this area as well. So we have to keep this collective embodiment of connective tissue in mind. The blood is also connective tissue, but it's not being formed by mesenchyme any longer. The blood-producing cells are already distributed to certain areas for the continued ongoing development. We saw the mesodermal cells originally with the blood islands differentiate into the blood-producing cells, and those blood-producing cells are distributed around and contributing to the formation of the blood. But we're keeping this idea of all the connective tissue coming from mesoderm, and now this special mesenchyme in the visceral arches that originated from the neural crest cells. So we recall also the pouch on the inside, the cleft on the outside, and the membrane in between. So the second arch is going to form the hyoid bone, the stapes, the styloid process of the temporal bone. So the cartilage within there is going to lay down the foundation for the formation of these bones, these sets of bones. It supports the formation of cranial nerve 7, the pharynx, stomadeum, the primordial mouth that we had talked about, the oropharyngeal membrane, and then muscles of facial expression. So there's a tremendous lineage of production of structures from the arch itself, and really is coming all from the neural crest cells, including the nerves that come into this area. And again, I know I say it every time, so I don't have to, but I keep saying it. The only exception to the neural crest cells is the alpha motor axon that is distributed to, well, any motor axons coming from neurons of the origin within the neural tube.
The motor axons, they differentiate from the neural tube, and the others are differentiating from where?
Neural crest cells.
Neural crest.
Yeah. So collectively, we're talking about all these things: connective tissue and neural tissue, and vasculature originating from the mesenchyme produced by the neural crest cells. But the only other neural component within here is the outgrowth process, the axon from neurons within what would be the spinal cord or the brain stem. These are cranial nerves, so it would be brain stem, of course. The third arch will form the hyoid bone as well, and will contain cranial nerve 9. So far we've seen cranial nerve 5 in the maxillary process, the first arch basically, 7 in the second, 9 in the third arch, and 10 is in the fourth arch. These also are cranial nerves that carry or contribute to the distribution of parasympathetic autonomic nervous system components. So the fourth arch, in addition to carrying cranial nerve 10, anybody know the names that go with the Roman numerals? 10 is vagus, 9 is glossopharyngeal, 7 is facial. So that helps remind you that the muscles of facial expression are being produced in the second arch, where we find cranial nerve 7. Cranial nerve 9 is going to innervate the musculature of the pharynx and the larynx, which form in the vicinity of the hyoid bone. And cranial nerve 10 is going to innervate larynx, pharynx-larynx muscles as well. So we now see table 10-1 from the textbook, and the details are given here. We have the arches first, the mandibular second, the hyoid the third, the fourth, and then some contribution from the sixth. And the next column is the nerves, 5, 7, 9, and 10. The muscles for the mandibular, obviously the muscles for chewing, the muscles of mastication, the muscles on the floor of the mouth, the mylohyoid and the anterior belly of the digastric, muscles deep within the ear cavity and in the back of the oral cavity, the tensor tympani muscle and the tensor veli palatini. So your challenge, of course, is to try to recognize any of these structures If you haven't studied human anatomy yet in great detail, you certainly don't know these yet. So give it your best shot. We won't overemphasize these. Excuse me. But you should be able to realize that the tensor veli palatini is somewhere with the palate. Very unlikely the hard palate. It's probably going to be the soft palate, which is in the back of the oral cavity. So it would be close to the junction of the mandible and the maxilla. So that puts us in the mandibular part. So you have to look for tips like that. The malleus and the incus bones. We know that I was catching a bit of The Simpsons with my son the other night and noticed that Bart's ears are still just about where they should be from the development of those otic placodes. Didn't quite migrate back far on his head. So it reminds us that the ears are originating in this general area. And we know the mastoid bone is just behind the mandible, and the external auditory meatus is right near that area. So mandibular, the first arch, obviously is going to contribute to the formation of the bones of the middle ear, and many of the ligaments in this area. Sphenomandibular ligament tells us right off, mandibular arch. The second arch, muscles of facial expression. So we are in the hyoid bone area, so the cells are going to migrate up into the facial region and contribute to the formation of the muscles there and bring along with it cranial nerve 7. The final middle ear bone, the stapes, and the muscle that is attached to it, the stapedius, are forming from this, the second arch, stylohyoid, the posterior belly of the digastric. So obviously this is information detailed to the degree that it is targeting first-year medical students that are taking human gross anatomy at the same time they're taking this course. Actually, these courses are, the embryology is built into the human anatomy courses at the health profession schools, especially medical school. So this is your opportunity to study it all at once. So I'm not going to continue to go through this list with each and every structure. You can take a peek at it. You should definitely remember 5, 7, 9, and 10, at least to match the cranial nerve by Roman numeral to that. And obviously, for skeletal structures, as we move farther down through the list, we should realize that as we get to the fourth and sixth contribution, that's when we should have the cartilage of the larynx. So the thyroid cartilage and the cricoid cartilage should be something that you recall. So this is figure 10.5 from the textbook, and it's allowing us to follow the development of the Meckel's cartilage and Reichert's cartilage in the first and the second arches. And we see the site of the developing internal ear right in this area, dorsal to the second arch, as illustrated on this figure. So the coloration is showing us the first arch cartilage, the second arch cartilage, third arch cartilage, and the fourth arch cartilage. So we see that the light blue is showing us the first two bones of the inner ear cavity from the first arch, and then the remaining middle ear bone from the second arch. The original Meckel's cartilage basically is part of the mandible, or no longer persists in that area. Excuse me. The third arch is going to contribute to the greater cornu or horn, the main portion of the hyoid bone. The lesser portion sitting above that, or on top of it, superior to it, is obviously coming from the arch above the second arch. So both the second and the third arches contribute to the hyoid bone, the greater portion coming from the third, the greater cornu or horn. And then the thyroid and the cricoid cartilage, the two I asked you to recognize as being laryngeal cartilages, are coming from the fourth in that area. So this is a view of the it's a ventrolateral view looking up into this region in a neonate with the skin and the connective tissue and the ear dissected away to the level of the middle ear cavity, and we see the incus and the malleus in this area, so they'd be coming from the first arch, very closely related to the mandible. The stapes would be down here lower with the styloid process, if they're not really showing in this area, that would be coming from the second arch. And then these are the muscles in the floor of the mouth, the mylohyoid muscle here and the digastric muscle deep to it. So this obviously is an area of very complex anatomical interrelationships of numerous structures and a tremendous amount of terminology. So you can't be expected to become an expert on this, so you have to strive for recognition and making some sense of where things are located. A real tricky one is the muscles of facial expression are obviously associated with the cheeks and the mandible, our lips and our nose and things like that, but they come not from the mandibular arch. They originate from the mesenchyme of the hyoid arch, the second arch. So they migrate up into that area and set up residence. And the cranial nerve 7 actually comes from below the ear and spreads onto the face to innervate these muscles. So if you can kind of picture the cranial nerve 7 coming around onto the face, arching from below on upward, it reminds you of the track that these mesenchymal cells have taken to get up into the facial area to form the muscles of facial expression. So where are the derivatives of the first and the second branchial arches? The first, that mandibular process with the mandibular nerve, part of cranial nerve 5, that innervates, goes into the mandible and innervates our lower teeth. The muscles of mastication attaching to the mandible. The mylohyoid, the anterior belly of the digastric, we saw below the chin, attached again to the mandible. That palatini muscle, the tympani muscle, and then the incus malleus and part of the mandible, and then a ligament that attaches helps to attach the mandible to the sphenoid bone, the sphenomandibular ligament. The maxillary process above forms the maxilla, the palatine bone, the vomer, the maxillary nerve, and the zygomatic bone. So it's contributing very much to the cheekbones and the upper palate of the oral cavity. The second has the facial nerves, muscles of facial expression, posterior belly, digastric, stylohyoid, stapedius. So it goes on through the list. So you need to work with that set of words there and build on your visual association skills and try to memorize as much as you can. Until you've worked with this information repeatedly through a variety of anatomy courses, it can't become a working part of your persistent knowledge. So this is your introduction to this terminology and the recognition that once you get into a health professions program and you're in the gross anatomy, you're going to be completely bombarded with volumes and volumes of terminology and trying to understand the relationship of all these structures. And there is a tremendous amount of effort that goes into that. You have to work with it, and that's only one of the courses that you'll be taking. So this introduction to that is just a bit of a teaser to make you appreciate just how much work goes into surviving the first year and each subsequent year, but the first year you kind of get used to it of a health profession school. So the pouches then, the first one forms the tympanic membrane or the eardrum, and the eustachian tube. So it pushes back in from the pharynx to form the pharyngotympanic or the eustachian tube and the eardrum itself. The second pouch forms the palatine tonsils that are at the back of our mouth. So when we've got I don't know if you've ever had it or seen it in a child where they open their mouth and you just see the palatine tonsils touching together at the back of the oral cavity. Those are the tonsils that are forming in that area. The third forms the parathyroid glands and the thymus, so that's down in the neck. And then the fourth forms the other set of the parathyroid glands. So obviously these pouches are extremely important structures. So here we see the pouches in this area. We see labels on the lateral tongue elevations. So we haven't even talked about the development of the tongue, but it's coming from this area. The thyroid diverticulum is between the first and the second arch, as we see here. So the thyroid gland wasn't on the previous list that we just worked with. It comes straight through from the floor of these arches. It doesn't come from any of the pouches. So here's the second pouch, the first pouch, second pouch, third pouch, and the fourth pouch. So these pouches, again, the first is the middle ear cavity, where the bones are in there that we talked about, the auditory tube coming into it. So the auditory tube, the eustachian tube, the pharyngotympanic tube, three names for the same thing. The epithelium of the inner surface of the tympanic membrane. So the tympanic membrane is covered by epithelium and it has underlying connective tissue. Second is palatine. So we start at the ear and we're kind of working our way around and then back through the pharynx. So we get to the palate, the soft palate where the palatine tonsils are located, and then we drop all of a sudden all the way down behind the thyroid. So these pouches meet the thyroid that grows straight down, and we'll see that a bit later. And the third pouch gives us the inferior, so the higher pouch, or lower numbered pouch, creates the lowest set of parathyroid glands, and the higher numbered pouch forms the upper or superior parathyroid glands. There's two sets of them. Let's not worry about the fifth one listed there at all. So without doubt, we can easily feel overwhelmed with this volume of information. The formation of this area is extremely complex. They are extremely complex areas. This is our interface with the world. Every organism has a highly ornate structure where it interfaces with its external environment. We interact with others of our own species. We interact with other species. We interact with non-living environment. So we pick up visual and auditory and taste sensations through the structures developing in these areas. We have the muscles of facial expression for interacting with our own species, with other species. We have the nasal cavity and the visual and auditory systems to orient to structures or organisms within our external environment, to determine whether they're food or not food, friend or foe, and respond appropriately and utilize those. We either bite to be defensive or offensive, or we bite to kill and eat. So all of these very important interactions with our environment, we orient to our environment. It's a tremendous area of an organism. The rest of the organism is a bit more simplified, so things are just going to get easier. When we get to the other end of the organism, things are still a bit complex, but not quite as complex. Tougher to get all that food found and chumped up and in than it is to get it out, I guess is one way to think about it, the food and the liquids. So let's continue on. We have to complete the discussion of this area and the formation of the face. I had tore out a page of magazine, Time magazine this morning, but I forgot to put it in my bag to bring it in. It's an advertisement or request for donations from individuals. I don't know if any of you have seen it. There's a team of surgeons that travel the world and repair, actually craniofacial surgeons, and they repair cleft lip, cleft palate in the underdeveloped countries around the world. Of course, there are two objectives that they have. One is to serve the world community that is disadvantaged and underdeveloped. The second is, just doesn't happen that much in a developed country, so you don't get the opportunity to develop those surgical skills. So the individuals that are involved in this benefit in two ways as well, from the reward of contributing to individuals around the world and improve the quality of their life tremendously, and also really to sharpen their craniofacial surgical skills. So, an understanding and appreciation for the development of the head and neck and face region is critical in healthcare. So the grooves, these are then on the outside. They develop during weeks 4 to 5. The first becomes the external acoustic meatus. So the pouch became, one of the things that contributed to was the pharyngotympanic tube, so from the inside up to the middle ear cavity. The cleft on the outside contributes to the external auditory meatus that comes up to the eardrum. On the other side of the eardrum is the opening of the eustachian tube. So we should be able to picture these things happening where they're just growing and coming together and having a thin membrane separate them and being related to the first arch. So the second through the fourth grooves are found in the cervical sinus. They basically will disappear. So the membranes where the pouch and the clefts come together, they're found in the floor of the four grooves. The first membrane becomes the eardrum, the tympanic membrane. So we see these membranes here. We see the cervical sinus developing from the clefts 2, 3, and 4. And we see the pouches undergoing tremendous changes, the third, the fourth, and the fifth pouches as they are migrating on to form their structures. We see the thyroid diverticulum forming here in the midline. It's going to push forward and down, and then these, the third and the fourth, that contribute to the parathyroids are going to come in and become related to that. We see the tongue buds beginning to develop in these areas also. So this first groove and pouch are going to come together, thin out, and contribute to the formation of the tympanic membrane, and we'll follow that on this series. So we see the first groove persists. It's modified in its shape, and it migrates into another location. It doesn't necessarily move itself. Everything's moving and getting bigger, and it becomes repositioned relative to other structures. So we get the external acoustic meatus feeding into this portion. So the external surface of the surface epithelium of the tympanic membrane is ectodermal origin, and the internal epithelial surface then would be endodermal origin. We keep our coloration here, the blue and the yellow, relative to germ cell layer origin. Connective tissue in between. Connective tissue is always mesoderm mesenchyme. We see the palatine tonsil forming from the second pouch, and the third and the fourth pouches continuing to grow and develop to form the parathyroid and thymus glands. We see the cervical vesicle basically progressing toward disappearing completely, and then finally we get to this stage where we see the tympanic membrane persisting with the external acoustic meatus and canal, and the auricle, the external ear, beginning to develop through elevations in this area. We see the foramen cecum is the term for the site where the thyroid diverticulum originated, and the thyroid is now down in the anterior region of the neck, anterior to the larynx, anterior and inferior to the larynx, excuse me. And we see how the parathyroids moved into this area. The palatine tonsils persist at the back of the tongue area. So we now can take a closer look at the development of the thyroid. It is the first endocrine gland to develop, and it begins to form in week 4. We saw it, the thyroid diverticulum, from the endoderm of the pharynx. So it's a core of endodermal cells. It's going to push down into the floor of this primordial pharynx at the base of the tongue, if you will, and move into the front of the neck region and migrate inferiorly. The connective tissue that makes up the thyroid gland, stroma, the vasculature and the neural bundles coming, well, let's not say neural bundles, the vasculature coming into it are coming from mesoderm. So we now introduce this idea of histogenesis and organogenesis occurring at the same time. Histogenesis are the tissues, all the connective tissue and the vasculature from mesoderm mesenchyme. We now throw mesenchyme in there to remind ourselves of the special mesoderm basically created by the neural crest cells. So the connective tissue makes up the stroma of an organ, the portion that holds it together, the basic foundation of the structure itself. The parenchymal cells are going to be the functional cells. So the endodermal cells of the thyroid diverticulum are going to move into this area and become associated with mesodermal mesenchymal cells that will form the stroma, and then the endodermal cells will differentiate into the functional parenchyma of this area. So this is going to be a common occurrence where we're going to ask ourselves from what germ cell layer did the parenchymal functional cells originate for this organ, and then we know the stroma came from mesenchyme mesoderm. So this is the game that we begin to play first here with the discussion of the thyroid gland, and as we continue on through the full development. So during week 7, the thyroglossal duct degenerates, and the thyroid gland assumes its position down in the deepest part of the anterior neck. So this duct should completely degenerate. It doesn't always, so there are some abnormalities. During the week 11, the gland assumes its normal histology of the follicles and the colloid responsible for producing thyroid hormone. So the parenchymal histogenesis is what we're talking about there. The stromal histogenesis is going to be occurring at the same time. So from a lateral view, we see again the first, second, third, and fourth arches, the pouches, 1, 2, 3, 4, and in the ventral midline at the base of the first with the junction of the second arch is the thyroid diverticulum. It's going to push forward a solid core of these endodermal cells, going to push forward, and as everything else grows and develops, it's still going to descend, but it's basically going to be left in its final position as other things migrate and move away from there. So it's a very dynamic process for the change in the shape and position of everything, and the the size of everything. So quite often we're picturing something starting here and just moving to a whole new location and then finally settling in. But again, we remind ourselves that we're starting small, so it moves to a location. Then as everything grows and expands and moves away from each other, it may not have moved very far originally, but the extension of its original attachment may have been stretched as things grew in size and girth, and other things came in to take their position. So we see the diverticulum. That original core of cells now has a little bit of a duct within it, an opening inside of it, and this is important to understand that when there are developmental anomalies, that is a key point to be made. The pit that is left, or the depression that is left behind, is called the, what did we call it on the previous slide? Anyway, the foramen opening into this area is the foramen cecum. It's killing me, I've got to go back and see it. Oh, the foramen cecum. Okay. So foramen is an opening. Now we see in this panel C that the thyroglossal duct is quite extensive as things have moved and done all the growing that we had talked about. The foramen cecum is still open, and the thyroid gland is beginning to take its position relative to the hyoid bone. And then the atrophy of the duct should begin in this area here, the future neck region, just above the hyoid bone. So here is a lateral view of the adult human, showing from the foramen cecum of the tongue all the way down through below the hyoid bone, and then a final descent. And we're making it appear, as I said, like it migrated this tremendous distance. But all these things were close together. As they moved farther apart, its trail was longer. But this is the full route of the extension. So it is coming through the primordial tongue, so that's the glossal part of this. It's going to end up forming the thyroid. So there is this little pyramid-shaped lobe on the superior anterior portion of the thyroid gland called the pyramidal lobe, and that is basically a persistent part of the thyroglossal duct. But otherwise, there should be no persistent component of the thyroglossal duct. It should disappear entirely. So that first pharyngeal membrane becomes the tympanic membrane. The blank gland is the blank endocrine gland to develop in the embryo. Obviously, that's the thymus and the third, the thyroid and the first. And the thyroid primordium consists of a solid mass of the endodermal cells, as we had noted. So the thyroid primordium consists of that solid mass of endodermal cells, and this aggregation later breaks up into a network of epithelial cords because of the invasion of the surrounding vascular and mesenchyme. So this is the point that we were making before. Let's think of the parenchymal cells separate from the stromal cells, so we can ask ourselves, from where did the parenchymal cells originate, in this case endodermal mass that is invaded by the nearby developing vasculature, which comes from mesenchyme and mesoderm as well, as the mesenchyme that's coming in to form the fibroblasts and reticular cells that contribute to the fibroblast, the collagen fiber and the reticular fiber network of the stroma of an organ. So this cytogenesis, histogenesis, organogenesis is coming to visit us again as a series of events involving different germ cell layers, different lineages of differentiation from totipotent to pluripotent to multipotent to bipotent to unipotent, and finally the fibroblast is formed, and it'll begin to dump out collagen into the extracellular matrix to form that extracellular connective tissue. And in this case, we're thinking of it as a stroma of an organ. Other cells would be reticular cells, that would secrete reticulin protein to make the reticular fibers of the stroma of an organ. So the next thing we want to take the last few minutes to discuss is the face, development of the face. So the stomodeum is the primordial mouth. We've mentioned this before. The prosencephalic organizing center is a very important collection of cells that appears and kind of signals to everyone, hey, let's get organized and produce this area in front of the brain. And the tissue migrated from the primitive streak and is ventral to the forebrain. So we have a lot of signaling going on to get organized and go through tremendous differentiation and development. There's a rhombencephalic organizing center ventral to the hindbrain. So this is reminding us of a segmental type of development still, but it's segmental but clustered so that we trigger the formation of an entire region or contribute to that triggering. So there will be 5 facial prominences. The frontonasal prominence that we saw before, it's unpaired, and it gives rise to the optic vesicles, the forehead, and the rostral nose. The maxillary prominences that we've seen will form the lateral part of the mouth. They are paired. And the mandibular prominences that are paired and caudal to the mouth. The active growth of the face is going to occur during weeks 4 to 8, so it's quite early on. Again, remember that this is a time period where individuals certainly should know that they are pregnant, but it's a very key time for increased nutritive intake to sustain this, and the tremendous susceptibility to any type of chemicals or molecules within the environment that can impact this. And so in underdeveloped areas, malformations or incomplete fusion of these structures is relatively common. So the nasal placodes will appear at week 4. They're thickenings of the surface ectoderm, and they are the primordial nose. So let's follow that just quickly in a couple of illustrations. Again, the stomodeum at 24 days from an anterior view, lateral view. The heart prominence, these are our landmarks. Rostral neuropore is not closed yet. And we're seeing the pharyngeal arches here. The first arches and the second arch are beginning to form. And from that prosencephalic organization center, we've triggered events so that we now get thickening of the surface ectoderm to form the lens placode and the nasal placode in these areas contribute to formation in eye and nose. You see the stomodeum and the rest of the first and the second or hyoid arches at day 28. Day 31, we see the nasal pit. The nasal frontal prominence is here, not labeled, but we know that that unpaired structure is migrating into this area. We see the eye, and where the maxillary protuberance or bulge is forming and meeting this portion, the lateral portion of the nasal pit. This is where the nasolacrimal groove is going to originate, and the nasolacrimal duct will be forming from that. We know that the nasolacrimal duct goes from the inside corner of our eye down into the lower part of our nasal cavity. So when our eyes tear up, little bits of tears, we might call it snot, but it's actually tear fluid that escapes from our nose, and that's through the nasolacrimal duct. So on an ongoing basis, there's fluid being washed toward the medial part of the eye and down through the nasolacrimal duct into the nasal cavity. So I'm going to stop with that set of slides and pick up there to finish the development of the face on Friday, and we'll proceed into Lecture 18. So as I had stated, we will have a lecture on Monday to complete this series of lectures for the exam. So even though the exam will be ready right after class, if you're not completely ready, you can wait until another time. So I appreciate your time and attention, and hope it stops raining, and if you have to go to another building, you don't get soaking wet.